Today's webinar will cover some metagenomic workflows in DNASTAR software. And before we get started, I'd like to just introduce you to our new website. If you haven't checked this out already, please do. It's organized in a very logical fashion, and you can see we have multiple different workflows listed here, and I'll just highlight some important resources. So for instance, this webinar is being recorded. For those of you that can't join us live today, you'll get a link to download the recording, but we'll also post this on our website under the training section. And there's some really nice resources here. So if you check this out, we have some workflows that include molecular biology and structural biology and genomics workflows, and resources like blogs that contain great information about a variety of different products. So again, if you haven't checked out our new website, please do. And with that, we'll kind of move on through the software. Just kind of a general overview. We offer software in molecular biology. And the molecular biology provides tools like phylogenetic sequence alignment, Sanger data analysis. Actually, I have a list here. Let me just get my highlighter here. There we go. There we go. So there's cloning and primer design and Sanger sequence assembly and RNA and DNA alignments. Agarose gel simulation is a new feature that we added earlier this year. So a whole suite of tools for molecular biology. And of course, we have protein analysis and structure prediction with some really nice visualizations. So we can look at 3D structures, make predictions on epitopes, and do structural alignments and folding and antibody modeling and protein docking. So it's a whole suite of tools that are really cutting edge and provide an excellent user experience. And of course, today we're going to be focused on genomics and transcriptomics. And in particular, we'll look at a metagenomic workflow that in our software actually has multiple different aspects to it. So we'll hopefully have some time to go through the different ways that you can look at metagenomics in our software. We also have, of course, resequencing and genotyping and de novo workflows and RNA-seq and ChIP-seq and some other workflows as well in this area. And so the software is quite flexible. And in a webinar like this, I'll have time to show you some of that flexibility. So the metagenomic workflows really cover a lot of different types of data sets. Sometimes it's aligning to a whole genome database, and so this database may be comprised of microbial genomes. It may have some fungal genomes in. Common repetitive elements from eukaryote contamination can be there, and so it can really scale up from just a handful of sequences that you want to compare a sample to a really large set of sequences. We can also align to 16S databases. So if you have your favorite subset or large database, we can do that alignment. There's de novo assembly. So in some cases, if your organisms aren't represented well in a database, we can do de novo clustering and identify what those individuals are. And then we have some nice tools that go along. Host removal. If you have a clinical sample and lots of the host DNA is present, you can remove that in a pre-scan. And then we can do some things like multiple sample comparison, and I'll show you some ways that we can do that, and compare relative abundance, try to figure out what's most abundant in the sample, or if it's changed between different conditions, if you have a changing population, and really identify some of those organisms. So, and there's different ways to do this in the software, so we'll spend some time there. So the data sets that I'm using today, there's a small data set. So this first data set is one of the early metagenomic data sets on the short read archive, and it's actually 454 data, and it's one that was used to detect a pathogen in human patients that had diarrhea versus normal. Of course, we're going to talk about poop today. It's metagenomics and lots of data sets for human fecal or stool samples. So we have this small data set. We'll go through a metagenomic workflow and align to a smaller subset of likely genomes, and then we'll take this data, import it through a data import wizard in our RayStar, which is a multiple set analyzing tool. So we can pull in any kind of data into a RayStar As long as it has a gene or a genome and a signal, we can make some comparison. So that's where we'll start, as a smaller data set. And then we have a larger data set that is really a very large clinical study. And it was to look at what happens to the human gut after treatment with antibiotics. So you would see the normal gut, treat it with antibiotics, which would wipe out a lot of the normal flora, and then how to best restore that. So of course, when people take strong antibiotics in particular, it can really clean the gut out and cause all sorts of issues. So it may be probiotics, or a fecal transplant is another, FMT, or just time. Give it a few months, and so you can look at the metagenome changes. And I just pulled a few samples out of just kind of randomly out of this really large study. So there's all sorts of other controls in different regions of the gut, and I just pulled a small subset so we could look at that for today's webinar. And with this, I'll jump out of the PowerPoint, and we don't want to keep those. And I'm going to launch Skema Engine. And so Skema Engine then is the starting point. And you can see I've got some development build here, but we'll go to version 15, which is the current released version. And what Skema Engine is we call it the wizard, and it allows us to set up our assemblies, and it guides us through the process of all different types of workflows. So it's really intended to be a guide that gives you some feedback and allows you to do different projects. And the three options are to assemble locally, and so that would be on my local desktop computer, or I can rerun an assembly. So if I run an assembly, it creates a script file. So I could rerun something I did 2 weeks ago by just loading that script file in. It'll populate this wizard with that all that information. So and they're always associated with the output. So they're always in the folder of the output, so you can always grab them and rerun things. And then we can also assemble on the cloud. And when we start talking about hardware, some of these data sets, the larger they get, in particular if we're going to be aligning to 3,000 to 5,000 bacterial genomes, there's some hardware requirements, and we'll talk about those briefly, but the cloud is really a nice option, and we'll set up a cloud assembly as well as a local assembly here. So we're going to start here local, and you can see that we have different workflows, and we'll pick the Metagenomic Workflow. And this will be reference-based. So you can see I have 4 options. I can align to a reference. I can first do a host removal, align essentially to a human genome, pull out all the human data, and then align the remaining data to my target genomes. And I can do the same thing with de novo. So some different options here. It just kind of streamlines it for you to make it easier. We collect some system information on your computer. And the one thing I do want to point out is if you intend to evaluate a demo of the software, the temporary file location is really important for the success when we're aligning to a large group of templates. And so that's why it points out how much space do I have. And generally you want, for these larger data sets, at least a terabyte of space. So a dedicated drive. And what that does is it writes temp files as it goes through all those templates, and it stores it on disk and runs the assembly and then erases it when it's done. And so if you're doing these larger data sets, either make sure that you have a temp drive or consider the cloud, which this is all just set up perfectly on a cloud computer. So now we're going to add the biome genomes. And so these will be the RefSeq subset. So for this first data set, I'm just going to grab, it's a small subset of likely bacteria in the human gut. And you can have sources, I've looked at databases, and there's all sorts of different databases now for human gut bacteria. And so it doesn't matter where you get the source, but this would be considered a small subset. I could run this on a laptop without any problems. The next data set will have, over 3,000 genomes worth. So this is something that's meant to go fairly quick. And now I know we don't see 454 data too much anymore, but it doesn't really matter. I could set this as illuminant, it would just be fine. So this is, we have 4 files, and you can see one is healthy, one set, and one set is diarrhea. So what we can do here is, in this setup, I don't have to set up each sample separately and run the assembly. It gives me some flexibility here where I can tell it, this is multiple sample, and I can group files together. So I can say, let's group these, and I'll just call this the sick group, and here's the normal group. So I'm just selecting these files. All right, and I could have, whole sets. There's ways to set replicates up. I could make replicates, and then with those replicates, group them together. So this is where we set up our experiment in terms of which FASTQ files go together, and sometimes you'll have multiple FASTQ files per experiment. Sometimes it's one per experiment, so it gives us flexibility for setting the project up. And now here's some of the assembly options, and I'll point out a couple of the really important things, and this is an alignment. So the expectation is the sequence data is going to align to genomes, and some of those genomes might be very similar to each other. We could have whole groups of strains that are almost identical to each other in our database, and so we don't know exactly how close they are. In general, if it's a large database, you're going to want to have a high degree of stringency. And so this minimum match percentage is set at 99, and that means that the read must match 99%. So if it's a 100 base pair read, only one mismatch is allowed. So it's really stringent. You could start a little lower, say 97, allow a little more mismatch. Really depends what your needs are. Are you interested in parsing out per strain, or is it just per group? Is that close enough? So it's the one parameter that you'll want to change. Go a little less stringent, a little more stringent, to see how that affects your results. So it's pretty simple. There's also some advanced option. I'm going to point out what the metagenomic workflow really means to the algorithm is all the way down here in repeat handling. And you can see it says place repeat reads all. And so in the case where I have 2 reference genomes that are identical except for one SNP, for example, really, really close, the reads are going to map to either one equally well. And so we don't want the aligner to align to one of the templates. We want to align it to both of them. And so we allow the aligner then to place the reads to all the templates that they match to. So depending on my database, I may have 1 million input reads, but if I have a highly redundant database, I could very easily have 10 million or even 100 million aligned reads. Now this is a small sample, so I don't expect as much of this kind of repeat handling. Now, if I picked once, all the other workflows that are not metagenomic, it'll say place reads once. A user could change it to never. You probably don't ever want to do that for metagenomic, unless you're just looking for those unique reads that make each reference unique from each other. So I guess there's a use case for that as well. But we typically go with all. And the reason that I'm pointing this out is that this is available in all the different workflows. And so I can go through, for example, an RNA-seq workflow and change this repeat handling to all, and I've essentially allowed it to be a metatranscriptome assembler. So I don't have to go through the metagenomic workflow to get a metagenomic type handling of the data. So it's 2 parameters. The alignment stringency, minimum match percentage, and the repeat handling are most important for this workflow. Okay, so we're setting things up, and I'm just going to call this, now I have this human. And so I name it, and I pick the output location. And let's see, I can go right here with it. It says it already exists. Well, let's rename it here. I've done this one, so I'll just add the number 2. So it's going to save these dot assemblies, which for the normal and the sick, which is a collection of BAM files and coverage files. And so I click next. So now it's assembling locally on the computer, and this takes about 1.5 minutes. So if you need to get a drink of water or something, or post a question, now is a good time. And so what it's doing here is it's we're watching the log of the assembly algorithm. And I can see streaming past, I can see the input data going in. And when you're doing these assemblies, you can see there's a button up in the right corner called Export Log. Anytime that you need technical support, say an assembly doesn't finish or you're having troubles with an assembly, you can export the log, save it to a text file, and then send that to DNASTAR. And we'll use that log then for any kind of troubleshooting that you may need with your particular assembly. And so log, now we're going to the second, so the first sample is done, it's going through the second. And you can see it says start aligning template. It's going through all the templates. So it's an extremely fast assembly algorithm, which is really nice when especially when you're trying out different parameters. You get results quick, you can look at them, try a different parameter, and move forward. So there we have process is finished. We'll have some options here, different programs to look at the data. So here's the output. It tells me, how many reads align, how many templates there were, and I can reveal the files if I want to download them and location. But the first thing I'm going to do is launch in SeqMan Pro. It'll ask me which project now I want to look at. And when I launch in SeqMan Pro, for some analyses, this is all that you'll need to do. If you're just kind of doing a rough calculation as to, what's in your sample, or a pilot run just to see, what's there, this can give you a really quick answer. And you can also use it to evaluate the alignment. Now, this is a really little data set, so there's not a ton of reads here. But what I'm doing is just expanding, and we can see in this window, these are all the genomes where sequence data aligned, and I'll sort by number of sequences aligned. And so this is a little clinical sample where you just pull a little sample or generate some data, and I can see very quickly, Bacteroides is at the top. So Bacteroides makes up, a third or a quarter of the human gut flora. And I see some others, and I see a Campylobacter. Campylobacter you probably don't want in your intestines. So this is a sick person. So that one would kind of jump out at me as saying, well, that is not something you ever want to see showing up. And then I see this Haemophilus as well. That can be a problem. So I see a couple of bacteria here that, while they're not at really high levels, you don't typically want to see in a normal gut. And so I could do the same, go back here. And of course, on a little sample like this, I can just eyeball it quick and see what's going on. And I often recommend that, even if you have a large data set, to run a little pilot, run 1% of the data or 10% of the data to see what's there. Sometimes there's, you'll see, if you align to a full database, a lot of human contamination. And so you might want to do a screen, or I'll just sort this one again, and I can see, well, I see some of the same here. I see the Bacteroides. I'm not seeing the Campylobacter. This is the normal gut. So I could see pretty quick that it looks like this small data set, the patients experiencing diarrhea had some Campylobacter present in their gut. And so it's, again, a real easy way to use the software. Now, if I want to have something more visual than that, you can imagine that if I had 100 genomes or 200 there, just looking through the list by the raw count isn't, or I have multiple samples, it's going to be more difficult. So I have another software here, and it's ArrayStar. And we'll let ArrayStar launch. And ArrayStar is, we have different workflows that are built into it, but it really also has a custom import kind of a wizard. And so the microarray workflow in particular really just expects text data in a tabular format, imported in, and allows the user to say, this is my gene, this is my signal, here's some descriptive columns or annotations. So it's a custom way to bring in data. So I'll show you another way that folks will do this analysis, is they'll just use this to import a file. And if I go to my output, let's see, where is that? Here. So what we just assembled is right here. And as part of the assembly, the dot assemblies contain the BAM file, so that's the alignments that we're looking at. There's also reports, and there's a per template report that's generated for these workflows. So if I have 10 templates or 10,000, I'll get a big table that gives me a count of everything. So here's that custom import wizard. So it's reading that file, and it looks like it's parsing it properly by tab elimination. So I'm just going to click Next. And this is where I get to define. So again, your data source, you may have data from some other aligner even, or some other analysis that you've done, and you want to make comparisons. You can still use this even if the data comes from elsewhere. And you can see these headers have pull downs, and I'm ignoring the first, and there I see template title. And in this case, the way ArrayStar works is it's gene-based. And I can tell ArrayStar, well, it's the entire sequence is the gene. You know, I'm not looking for individual genes in this scenario. Sometimes I am in a metagenomics, but in this scenario, I'm aligning to the whole template. So the gene is going to be the whole genome. And then I have raw count, seq count, as my signal. So just two things. I have template and count. And so I'm just going to finish that. And I'm going to do that again. And so this is a way to do that. And it recognized that was the same format. And this is linear scale. I'll just bring this in. And I can get this the name. So it's looking at the experiments, and I can say, well, let's make this a little smarter. And so I can highlight the part of that name, and then it comes up in normal and sick. So there I've got my two groups in my experiment, and I can go to a gene table and get a side-by-side comparison. So I can see there's normal versus sick. And now if I want to get a nice picture, we're doing all this just to get a nice picture. So I can cluster this and get a real simple little heat map. And the heat maps, of course, look really similar. And here's our Campylobacter. We see that I know it's a small font, but the sick individuals are on the right. And we can see the patterns aren't the same. There is a little bit of difference here. And of course, it's because we have Campylobacter and the Haemophilus showing up in the sick individuals, and they kind of pop out with a heat map. And so that's the really kind of the basic overview of doing the metagenomic alignment, looking at the alignment, and then going to ArrayStar to import that data and generate a nice image. And I did, so we'll close this. I am going to go back to the alignment. I forgot to show you one thing, and that was we can look at this aligned data. And even though it's really sparse, right, we only have 4,000 reads here, I'm going to go to the strategy view. And the strategy view is a map then that's going to show me coverage. And again, this is also, I call it kind of a sanity check. And for example, if the reads, and you can see this coverage, there's little sparse kind of spikes here, and I'm going to double click there. And what that means is I've got coverage across this genome. So there is some sequence, and there's some sequences. And I can see just two aligned sequences, and here's the Bacteroides genome in this gene. I can see one mismatch was allowed. So this one read was allowed because it wasn't 100%. And I can get a feel for how stringent my alignment is. And so as you change the alignment stringency, lower or raise it, you can look at the alignment and make sure that things aren't too stringent or not stringent enough. So it's helpful to sometimes look at these. Now there's also we can look at that output in an Excel sheet. And so let me just, and so there's more information there that you might go through. And so I'm going to go back into the reports, and I hope Excel is, there we go, on this computer. And so I'm going to also take that same report and look at it, and there's some more information here that's useful. And it looks like this one was actually cleaned up a little bit. So so what's most interesting here is I get my template ID. So there's the name. I get the seat count, but I also get template coverage %. And what that means is how much of the template is covered by aligned reads. And as we get more and more data, much larger data sets than this, you'll see that sometimes the coverage is, a particular bacteria can be covered by 90% or 80%. And so you can use that in addition to the count to determine the relative abundance of something that's in a sample, rather than you might have some conserved areas where the reads tend to pile up and the template coverage is lower as a %, even though the seat count is quite high. So this per template result table is also very useful. Okay, so let's go to a larger data set now. And so and this larger data set, we're going to do a cloud assembly. So if I determine that I don't have much of a scratch disk, or I don't have one that I can dedicate, I'm going to choose to use the cloud. And the cloud is something you can ask a sales rep here. When you're trying out a demo, you can tell the rep that you'd like to try cloud assemblies. And this will use some of the new Amazon computers. They're fast, they're cheap, the data transfer is quick. And so the way that you do this is you select upload data, and there will be a login for you, or it'll just be your demo user account, and you log in. And this is a cloud data drive, which is a really fast data transfer. So you can see I've got some, these are some of my demo data sets. And what you do is you create a folder like that. And I can upload using these icons. I can upload a folder or upload a file. Here's a couple little, and you can see that's over 9 gigs of files. And so this will very, very quickly move your data up to these folders. And you can monitor the progress of it. This is actually going to be overhauled. This interface, we'll have a release a little bit later this year that updates this cloud data drive and makes it a little more user-friendly yet, and makes it look better and perform a little bit better. And so please check that out later this year. So that's how we upload. And once you have data up on the cloud folder, then we can run an assembly. In this case, I'm going to pick the transcriptomics RNA-seq workflow, because I'm going to try to do some of the quantitation up front of the aligned reads. And we'll use that same repeat handler that I showed you earlier, where I can switch that over to all, and it essentially becomes a metagenomic type workflow. So reference-based, what I did is I added all these. This is well, I guess I can show you here. So when I click add now, it's looking on the cloud folder, and it's a microbial genome database. And so this is just FASTA files, A through Z from NCBI. So it's about 3,500 genomes. And so I selected those. You can see they're zipped up. They have zip extensions. And we're going to use each one of these reference sequences as a gene. So these aren't annotated, so there aren't quantitations against particular genes, but we're just going to use the whole reference and quantitate against that. And so now here's all the data. So again, we added data. And the data that I uploaded was for this this human gut antibiotic. So this is all brought in, and the different groups then contain different pieces of information. So I've already said that this is actually multiple sample, and there's replicates, and I've named the replicates here. And you can see it's just the file name. And now the next step is to group them into experiments. So this is, again, the interface with a bigger set of samples where I name the replicate and now I group them. And I don't actually have I don't remember the exact numbers. I'll just show you how you do this. I just select it, and we're going to group them, and this is going to be our control, our normal. And then this will be antibiotic. So human gut right after antibiotic treatment. And group these, and FMT, fecal transplant, to try to reestablish the biota in the gut. And I'm just going to select these, group. And this was probiotic treatment. And then finally, no special treatment, just lots of time. I don't recall exactly how long. It seemed that it was several weeks' time. Does it just reestablish on its own after the? So that's how you set up a larger experiment now. Replicates with multiple, and it can be, there's no real limit to how big this can be. It can handle a lot. And we're going to select a control. And for some comparisons, identifying the control up front will matter. And we can see our match percentage now is lower. So I'm going to set this back to 97. And important thing to remember is I want this as metagenomic, not an RNA-seq. So I'm going to change this. And by switching that to all, this becomes a metagenomic workflow. And I can normalize. And this is using RNA-seq type tools to normalize. And so there's different options here. I'm not exactly sure which one will work best for all the different experiments. DE-seq2 is for differential, compare experiments to controls. And people are just starting to kind of look into doing that for metagenomic data. RPKM has been around a while. Or I could say none and do some of this down in RayStar. Doesn't matter. I can just select none if I want. And we'll just name it human gut antibiotic meta. And pick the output folder. And again, this is going to go to the cloud folder. So I usually make an assemblies folder. And this is really nice too. With this many experiments, one of the big advantages is it can run them all at the same time. So the way the cloud works is that you see all these for each sample here. If each one takes 2 hours, for example, doing that locally on a local machine, 2 times that many experiments can take a while. So when I run in the cloud, it'll spool up a separate cloud machine for each sample. And so you can do these large data sets in a much, much faster time. And the cost is really almost negligible. It's gotten so low. So, you can certainly try out some demos at no additional cost. So here is assemblies ready to begin. There's the script when I start it. And what it did here, it says upload succeeded. It's uploading the data map and the script instructions. And it'll take a little time here. And what it's going to start doing is looking for cloud computers. So it's sending that data up to Amazon. And so it'll spend probably 10 minutes or so loading these all up. We'll come back and look at it and see where it is. So it's not too exciting watching that. So once it's done assembling, there will be, because I ran this through an RNA-seq experiment, there will be an RayStar project ready to go. I don't have to use the custom data import wizard. So it'll be in RayStar already. There'll be a .astar project that I can download then. And those are relatively small, so I can just download that project and start my analysis. If I want the assemblies, I can download those too. Those are a lot larger. They have BAM files and aligned reads. But they'll download it. This may take 30 minutes to an hour to download all of them. But what I did in this case is just the RayStar. And nope, not that one. The other one. So this is that project then. And let's see if I can make this. I'm going to make this a little bit bigger here. So here is a RayStar with different. So we don't need that one. The experiment setup. So I can see that I've got the control, antibiotic, probiotic after post-antibiotics, the FMT, post-antibiotic, and time and recovery. And each one of these then is a replicate, essentially, experiment. And now I can start to, to look at and there's all different ways to look at these. The first place that we want to go is the gene, called the gene table, but it's really the list of all my genomes. And you can see in the bottom left corner here, there's about 3,500 different genomes here. And this database from NCBI also contains lots of human repetitive fragments. And there's some, like here is Aspergillus. There's some common eukaryotic and fungal genomes and repeats that show up in metagenomics. So they're in here as well. And I can remove them, if I don't want. I see all these Aspergillus, right? Now that could occur in the gut, but if I say I don't want those in my analysis, I'm going to select it and remove the selection from my view. So this is a it's not like an Excel sheet, but it's a multi-state table. So I can have things selected with check marks, and I can reduce the size of the table by, and this is an important piece of ArrayStar, only showing the selected genes. That's the genes that I'm interested in and working with. And so one thing I like to do is just first kind of look at the data before I filter anything. Just see what we're dealing with, and you can see here's the raw count. So this is the control, antibiotic, probiotic, fecal transplant, and time recovery. So the thought is that the normal gut will have lots of diversity. The antibiotic treated, the diversity should be reduced. And what's unknown is how effective are the three treatments, probiotic, FMT, or time recovery, and how do they compare to each other? And now I only grabbed a few samples from a huge clinical trial. So I know that there's a lot of sample-to-sample variation depending on the individual that's involved. Everyone's got different populations in their gut. So the best way to do this would have, everything here. But for today's demo, it would have been a little bit too much. But I can sort these columns. And what I did is just sorted by the raw count of a line reads. What's most abundant, in the control sample? And you can see right away we have our Bacteroides, a couple different species of those right at the top, which makes a lot of sense that we'd expect, that to be the most numerous in most gut samples. And as I look across, I can see, whoa, the antibiotic is reduced by quite a bit, but then this probiotic is really low. And we have this post-antibiotic FMT control. Looks like stuff has come back, and then time recovery, it looks like the numbers get bigger. So you can kind of see a general trend where the control, high count, time recovery over enough time, high count, and kind of this dip in the middle. And as I scroll down, I'm looking at some of these other columns, and as I look at, say, probiotic, you can see that you're purposely ingesting different types of bacteria or yeast, and some of those kind of pop up. I can see them as I scroll down where some things start showing up, that like this lactobacillus, you see it show up in the FMT control. And so certain things that you would expect to be in a probiotic kind of will pop up in some of the samples more than others. Okay, so that's kind of the raw data. So let's just look at the whole thing. So if I select the entire table, well, let's make a cutoff here. I'll make a cutoff of a raw count of 25 or so. So I'm just going to select this part, and we're going to cluster. And I can see lots of different kind of pattern here, but let's zoom in a little bit. And, oops, too far. Here, let's see. I want to reset that one. I forgot to do one little thing here. So let's select just those. So this is 611 rows now. And we're showing only those genes. Now we'll cluster just that subset. There we go. Okay, that looks better. So it's just those top that have enough signal in the control. And you can see that the overall pattern, a lot of these are still at kind of low level. Maybe some of the noise region, you know, where we're getting some map reads. Down here at the bottom, you see very much some differential here and these are the higher expressed or the more abundant bacteria that were in the sample. And so control is the first column, and the darker red indicates, you know, more signal there, more aligned reads. And as I expand in, you can see in the far right that there's our Bacteroides again. So we've got, you know, this cluster of species here of Bacteroides represent a large part of the gut. And if we just focus on those for now, we can see that in the control, it's red. The antibiotic, which was measured pretty soon after the antibiotics were administered, it's reduced. It's a lighter color. And then really our treatment post antibiotic with probiotic, pretty weak. Same with FMT. And then over time, that looks pretty close to the normal. And so the treatments really didn't replace some of those, you know, right away. It really took just time in those cases. And as we look down here a little bit further, we see that there's, you know, some that got wiped out completely in the antibiotic treatment. And you can see, you know, as we go through here, a number of bacteria that just disappear the signal. And these blues then and light yellows indicate, you know, reduced diversity in the gut. And then as we go, thank you, everybody. Again, if you have questions about metagenomic workflows, feel free to contact DNASTAR. We'll have the webinars up on our website. And also, if you've registered for the webinar, you can download that as well. So with that, thank you again, and I'll answer any questions that have come up.
So the first question is, can Sanger data be used in metagenomic assemblies?
Yeah, I showed 454 in Illumina data. We also have the capacity to take Sanger data, align it same way. Sometimes the metagenomic Sanger data is a forward and reverse read that's paired together. We can do that and create really accurate consensus sequences from those, and then use those to do the analysis. So yes, both NGS and Sanger data is supported.
What are the sources of metagenomic databases, and what format do they need to be in?
Yeah, so the databases are, you know, so of course FASTA format is accepted, GenBank format, so the regular sequence formats. Depending on the format of the database, you may need some editing on, for example, the FASTA headers. So in the examples that I used, to make things easier, we wanted to see the species names, you know, on the on ArrayStar and SeqMan, so I can quickly determine and do my analysis without having to look up you know, a database ID number. And usually those modifications are just pretty simple in a text editor. And so that's something that we help our customers with all the time. If you have a database from, you know, Green Genes or some other 16S database, we can help modify the headers so that your analysis is a lot easier. You know, the names show up that are human readable. So again, it's something that we do with our customers all the time.